name is Michelle Ponto, and welcome to KAUST Live. Today, my guest is Gustav Paulay from the Florida Museum of Natural History. Gustav is the curator at the Florida Museum of Natural History, and he's here today as part of our Fall Enrichment program. Thank you for joining us today.
Thank you, Michelle. Glad to be here.
So your role at the Museum of Natural History is that you are the curator of invertebrates.
Yes, I am.
Tell me, what are some of the collections that you manage?
Well, invertebrates are basically almost all animals on the planet. You know, people think of animals as mammals and birds and reptiles and things like that, but those are all vertebrates, which is, if you divide the animal world into major groups, there's about 30 major groups called phyla, and all of the vertebrates are just one unit in one of those phyla. So everything else is invertebrates. So the corals, the worms, the crabs, the sea cucumbers, the sea squirts, bryozoans, things you never heard about, and so forth.
And you're here at KAUST this time specifically because we're doing something with biodiversity in the Red Sea.
Yeah.
So how is your collections related to that?
Well, to understand biodiversity, you have to understand the animals and the plants and other organisms that make up an ecosystem. That's what biodiversity is about. And so that's the science of taxonomy. It's telling species apart and understanding who they are. It's sort of like knowing what a dog is or a cat is or a rabbit is. And those are obvious, but when you get out in a coral reef or any other complex habitat, there are thousands of species out there. And to learn about them and to study them, you have to take samples of them and then determine who they are, analyze them both in the way they look and their genetic makeup. And museums are collections where we put specimens like that so we can compare them from different places. They're basically a library of life. So you can have in a single place comparative material from many places, and from that really understand what the species are in particular ecosystems. So they're an essential tool for studying biodiversity.
And how do you get the samples? Do you go out and get them yourself, or are they donated to the museum? How do you get them?
They come in a variety of ways. So of course, we do a lot of field work ourselves, and I've been at KAUST several times on expeditions with the Red Sea Research Center with Mike Beruman's group, and we help them collect samples, and then we can help them determine what they are. Or, you know, if people have problems identifying their samples, they can send them to us, and then we can study them there, or you know, museums are also clearinghouses where we engage others who have expertise in this material and get them to help identify samples. So if somebody is working on a complex group, very commonly they are involved with a museum collection for their study.
Okay. And how are they archived? For example, you have your physical museum, but say I was a researcher in Japan and I wanted to see what you guys had to compare. How would I be able to access that?
Well, as a first step, all of our collections have been cataloged, and their identity, where they came from and so forth, are available in a database that's accessible online. So the first thing you would do is you would search our online catalog and you would find out whether the organism of interest is available. And after that, you may be able to get some additional information, like the photographs we take of these animals alive are often up on that database as well, and you can look at them. Sometimes if they have some of their DNA sequenced, the DNA sequence data may be accessible to you once you know that there's a sample that's been sequenced. There are other databases that hold the sequence data. And then finally, if you're a more serious person or you're really curious about these samples, you can either ask it on loan, in which case we would, free of charge, package it up and mail it to you if you're a bona fide researcher and have expertise, or you could come and visit it and study it there.
Okay. What are some of the challenges you face when identifying different species when they come in?
Well, some animals are pretty easy. It's pretty easy to tell a cat from a dog, but other animals are much more difficult, and the expertise is really limited. We had one of the Fall Enrichment speakers just at noon was talking about soft corals, where she was holding up her hand as to the number of people on the planet who can identify that group, and there's about 5 of them. And these are animals about this big, and they cover large chunks of reefs, and yet the expertise is so limited, partly because they're very difficult to work with. So in those cases, you would have to do a pretty detailed study. One of the emerging tools that really helps people is getting DNA sequence data from a lot of these animals. So in groups that are challenging to identify visually, you can have the DNA help you, and then we would take samples from the organism for that and sequence it and have it identified that way.
Okay. Yeah, I know you're going to be talking tonight about more of these tools that you'll be using, and one of the things that I was reading about is the importance of the biodiversity surveys. What exactly are these surveys, and why are they important?
So if you go out to a typical reef right off here of KAUST, and you look at that reef, and it's going to be just incredible and full of color and fish and corals and everything, there is an immense amount of diversity on that reef. One of my friends, David Lichwager, did a photo shoot of 1 cubic foot of coral reef. He's a National Geographic photographer, and what he does is he looks at 1 cubic foot of different habitat on the planet. And he took 400 different organisms out of that 1 cubic foot and individually photographed them. That's how many he found in there, probably 200 species. So in order to study something like that, it's just a huge amount of work. You cannot just, if you want to know all the diversity, you cannot just go in there and look at the fish and think you're done, because then there are worms and then there are crabs. So what large-scale biodiversity surveys do is they try to cover as much ground in terms of the organisms as possible. So on a reef off of KAUST, a typical reef would hold 3,000–4,000 species. And to collect all of that is really an enormous task. But what a large-scale biodiversity survey does is it focuses on a system like that, and with a lot of sampling and careful, looking for organisms, you can cover 1,000 or 2,000, and then you can get a really broad understanding of that diversity instead of just the typical 50 or 100 species that most surveys would do.
Are these surveys still mostly done through just human interaction, or is there technology that's involved in gathering the data in the sea?
So a lot of it is, the traditional surveys are basically human collecting of physical specimens, or you might bring back a bucket of rubble or sand and then extract the specimens back in the laboratory. But one of the emerging tools today is to do it through just the shed cells that organisms produced. And this is the technique of doing environmental DNA sequencing or community DNA, depending on the method you're using. And you can either brush off pieces from the reef or take water samples from the reef. And we're all shedding stuff all the time. You could go, and there was a great movie, Gattaca. I don't know if you've seen it. We are shedding DNA everywhere. So is the reef. So is everything on the reef. So you can get an idea of what's there from the DNA. And that's a very powerful method. But there are still a lot of questions with the DNA. It's a very new technique, and it picks up things that you don't want to see. For example, we have pseudogenes, fake genes in our genome that are not the right thing. And so it can pick up a lot more diversity than is really there. So one of the major intersections of research today is to get DNA data from individually identified species to really understand them. And by that, you can better interpret the eDNA data that we are generating. Right now, the eDNA data is still a lot of people have reservations about it. There are clearly biases and problems with it, but the more we understand about the source of the DNA, the better we can make use of the eDNA that we can get by these other methods.
So if I was a marine science student now, or a biologist, I would want to have that biology background, but also have some knowledge in the extreme computing side, because that's going to help me.
Yep, exactly. And there is this dichotomy in our field, really, that if you don't understand the organisms, you're going to be lost. If you don't have the high-tech molecular and bioinformatics background, you're also going to be lost. You really have to have both. Somebody who has one half of that is missing a very, very important part. Or you need a team that brings that together. But those are, right now, really essential components of this kind of work.
And what's happening with, say your collection is 50 years old, the stuff that was 50 years ago didn't go through DNA sequencing.
Right.
Do you go back and look to make sure it's correct, or how does that work with the new technology combined with how things were done in the past?
So one of the things that DNA allows us to do, first of all, is reinterpret the old taxonomy. So often we find that either the old taxonomy is right, and you can just do it by looking at the organism, or you may find that it may not have been right, but you found a new way to look at the organism, so you can now tell it apart. And instead of looking that it had 3 bristles there, now you look at that cross-banding pattern on it, and that's what tells you what the species is. So you may be able to go back to those specimens, very often you can, and tell them by classical morphology, and it's as usable as it always has been. Also, many of the older specimens are sequenceable. I mean, we are sequencing Neanderthals, right? I mean, 50-year-old museum specimen is not that challenging if you really want to have that data. But beyond the identification, those specimens are there for many other kinds of study. I mean, we do morphological work on them. We do comparative analysis of size and structural patterns, intraspecific variation. You can even get isotopes out of them that tell you about past conditions. So one of the heavy uses of our molluscan collection is to get shells that were taken pre-bomb, meaning before the first atomic bomb tests were, to look at the ratios of carbon-14 in those shells, because you need that as an environmental signature for that area to interpret C14 data. And you cannot do it with modern collected materials because they've been contaminated by the fallout from atmospheric atomic bombing tests.
That leads into my next question. What are these collections used for besides identifying new species?
A million things, yeah. They are really just, we have a huge number of visitors. They're used for setting baselines in isotopes. They're used by archaeologists to interpret old human cultures, because paleo collections of identified remains are very important, but to identify them you have to compare them against something more whole. They're used for looking at evolution of all sorts of features. They're used for just looking at phenotypic variation, relationships of organisms, biodiversity, and so forth. It's a very diverse field. One of the grants that is very big at our museum right now is called OVERT, and what it does is it CT scans specimens. So it's focused on vertebrates, but people could do it on any kind of taxon, and you do these three-dimensional scans of rare things and common things. And now we are generating just gobs and gobs of three-dimensional reconstructions of the entire body of these animals that you can get online. So you can see that's precious specimen, and anybody can go and rotate it and look at it from different angles and ask questions and test hypotheses with those.
How quickly does a certain area change? For example, say you did a biological or biodiversity survey in the sea close by where you are. Mm-hmm. If you went back 5 years ago, would you find completely different things, or would it be the same things there?
If you asked this question from me 20 years ago, I would have said it's the same. There is variation, especially in the ocean. Some animals come and go on cycles. They are there 10 years from now and they disappear for 5 years and so forth. So there's a little bit of that kind of variation, but on average things stay around. Right now things are degrading so fast that it's really horrible. And one of the things that really motivates me in these surveys is simply to record what's there before it's gone. To give you an example, we were down in the Khorasan Islands in 2013. Some of my friends and colleagues went on a different expedition in 2014, and there was an expedition there last year. In the first two expeditions, it was an amazing area. We got a ton of very interesting material and have studied it, many new species and so forth. The last expedition found 80% coral mortality, and the reefs got totally trashed, and we don't know how much is left at this stage. There has not been a biodiversity survey to look at that, but clearly we got a lot of things that we may never be able to collect again there.
Wow, that's devastating.
Yeah, it is. It really is, and that's why I'm sort of this is my life and my passion right now, is these large surveys, because if you don't record it today, future generations will never know about it. And it's just like seeing a dodo or a dinosaur and being there with it, knowing that 50 years from now, 100 years from now, our records are going to be what people will know about this place.
And in your opinion, why is the degradation happening so quickly in some areas?
It has to do with the climatic and circulation patterns of the ocean, which I don't understand well enough. The ultimate cause is global warming and climate change for reefs. Reefs are incredibly sensitive to overheated water, and the consequences are devastating. And I've been working on this for decades, and I've seen enough of it that it just breaks your heart. Yeah.
Okay. I'm going to see if there's any questions in the audience from anybody. No? Okay, well, I have one more question.
Sure.
How did you get into this field? Because usually when you're in university, you're thinking of, I'm going to do this. Were you thinking to be a curator at that time?
No. When I was maybe 7, 8 years old, I read Dr. Dolittle, and it fascinated me. Talk to animals, travel inside snails, see the world. And from that point on, I knew I wanted to work with nature, be a naturalist. And it ended up being working on marine invertebrates, but it could have been anything. My initial passions were mammals, and then reptiles, and then fish, and so forth. And I'm interested in everything. And working on invertebrates reflects that, because I am working on everything. I ignore the vertebrates simply because there's so many people working on them. There's no reason to not to waste my time, but to spend time on it, because they're going to get covered well enough as they are. but the rest of the biosphere, there's very few people studying it.
Okay. Well, that's all the time we have for today. Join us this afternoon at 5:30 for Gustav's keynote on how little we know about the coastal biodiversity and how to change that. We'll be streaming it live on Facebook, or if you're on campus, join us here in the auditorium. Thanks for watching and thanks for joining us.
Thank you, Michelle.